Good evening, everyone. Am I audible?
Yes, ma'am. Good evening.
Am I audible? Okay.
Yes, ma'am.
Screen is okay. So previous up to the cell biology and mitosis meiosis are over. So sir has given brief idea regarding the Mendelian genetics little bit. So today we'll be discussing regarding the Mendelian genetics. So before starting Mendelian genetics, some terms are important which you need to know. So I'll be explaining the terms briefly and quickly. Okay. So tell me regarding the what do you know about a gene? What is a gene? Yes. What is a gene? Anyone? Gene is a stretch of DNA which is having means which can be further which has the information which can be further decoded to form the RNA and subsequently the protein, which will have some sort of function in the cell and it will give rise to some sort of expression in the organism ultimately. Okay, so that is a gene. So gene is nothing but a stretch of DNA which is having a particular coding sequence within it, and which that codes have a particular meaning carrying with it. If it is not that it is simply a stretch of sequence, but it is a set of codons which are actually having some meaning, which can be decoded to form some sort of proteins, which will ultimately give rise to your phenotype, right? Then what is a genome? Any idea what is a genome is?
Complete set of gene present in an individual.
Yes,
Some
complete
total
set
of
of
haploid chromosomes in an
yeah.
individual.
Yes, complete
Complete
set
set
of
of gene.
gene that is present in haploid individual, that is called as genome. Okay, because whatever genes we carry, that are present in two sets in diploid organism. So whenever we are talking regarding the genome, that is the complete set of gene that is present in the. Please mute yourself. Whoever is unmuted, please mute. Whenever I ask the questions, please unmute and answer. Okay. So the genetic or gene content present in an haploid cell, that is known as one gene, one genome, and that's why all the diploid organism have two sets of genome with it. Okay. So next is locus. Locus is the exact area, or it is the position where we can find the particular gene. Okay, to illustrate that thing, I have taken two chromosomes. This is the chromosome that is homologous pair of chromosome. This is unreplicated chromosome. Whenever the chromosomes are represented this way, means one chromosome is represented like this, another chromosome is replicated like means mentioned like this. These are two homologous chromosome, but they are unreplicated in condition. If they are replicated means in commonly very often you would have seen in the books they have mentioned like this. So whenever you find this sort of denotion, means one chromosome is carrying four things, means two chromatids separately, these are replicated ones. So here the chromosome I have represented, these are unreplicated chromosome. Keep that in mind. Okay, this is unreplicated chromosome. Okay, so in unreplicated chromosomes, all the unreplicated chromosome have a particular location where the particular gene will be found. So wherever this particular let's say this is the flowering color, gene for flower color. So wherever the particular gene is found in this particular chromosome, and at the same location in the homologous chromosome, you can find the concerned allele present in the other chromosome also. So both the chromosomes, homologous chromosomes, will carry the particular gene at a particular same position only. So that is one of the point. And since I am telling that these two are means this is the replicated, this one is was originally present and this one is the replicated one. So if here the flower color gene is present, means here the same allele will be present. Okay, and if here one allele is present, means in the replicated part same allele will be present over here. Let's say here the purple color, like here they have mentioned, if here the purple color allele is present, means in the counterpart here, in the another chromatid, the same purple color allele will be present. And here if the white color allele is present, means here white color allele is present, here in the counterpart of another chromatid also white color allele will be present, right? And these four chromatids will be ultimately segregating into the four meiotic, means four gametes that are produced from the meiosis event. Okay, so each of the gametes will actually carry one chromatid. In that condition, that one chromatid will ultimately represent one chromosomes, and they will be representing like this, and they will be carrying, means they will be representing the one chromosome, that is unreplicated chromosome, like I presented over here. And after that they will be growing for interphase again, and they will do the DNA replication, and again the DNA content will be doubled after that. Okay, so that is what happening over here. So here the point to remember is that the homologous pair will have the same allele at a particular position in both the chromosomes. Correct? Then alleles or allelomorph. This term tells about the alternative forms of a gene. Every one of you might be knowing, and they occupy the same position or locus in the homologous chromosomes. The term was coined by Batson and Saunders in 1902. Okay. Then coming to the two types of trait, that is dominant or recessive. If when we can call a trait as dominant, when we can call a trait as recessive, when the trait is expressing wherever it is present, whether it is in homozygous condition or heterozygous condition. Means homozygous condition means both the alleles are same. If the both homologous chromosomes are carrying same allele, that condition is called homozygous. If they are carrying different allele, that then we'll call this as heterozygous condition. So the allele which can express itself both in homozygous and heterozygous condition, that allele will be called as dominant allele. Okay, and it has the ability to suppress the contrasting allele, that is the recessive one. Then what is the recessive allele? The allele that does not express itself when present with the dominant allele, and it has the ability to express only and only when it is present in homozygous form. That is, only the recessive allele is present in both the chromosome, means it will express itself. That sort of allele is called as recessive allele. Okay. I already told you what is homozygous and heterozygous. So Mendel has used the term pure for homozygous, and Mendel has used the term hybrid for heterozygous one. Okay, that this is the information you need to remember, like what terms Mendel has used, because by that time Mendel has given the laws and everything. By that time these terms homozygous, heterozygous, these things were not there. Okay, so for homozygous thing he has used the term pure parent, and for heterozygous he has used the thing that is hybrid. Okay. Then here I have highlighted something, that is homozygous organism when cell pollinated or inbred always give rise to true breeding or homozygous forms. So whenever you see something called one sort of progeny only, okay, so there is nothing, means there is no segregation pattern you can see when you are breeding, when you are selfing a particular homozygous individual. So that's why they are called as true breeding, means always and always they will give rise to the progeny which is similar to them, exactly looking alike to them. Then heterozygous individual, for a for a zygote or a diploid individual developed from it carries a pair of allele. Means a zygote or individual which is having the particular thing, that is heterozygous condition, that may carry different sort of alleles in heterozygous condition. Means the progeny of heterozygous can be homozygote also, heterozygote also. Okay, like we see in the case of a heterozygous condition, for simplicity we can tell that the heterozygous condition is capital A small a. So if you just self the particular thing, the capital A small a, you will get all three types of progeny. So you will get capital A also, capital A also, small a also. So all the types of progeny you will get from heterozygous individual. Now the important point here comes is hemizygous individual. What are hemizygous individual? When the genotype of the determined by single allele in the diploid condition. Yesterday somebody was asking regarding the hemizygous condition. So hemizygous condition is like whenever in this sort of condition from let's say up to this much, this region is deleted because of some reason. This portion is deleted because of some reason. This portion is lost. Deleted means this portion is lost. So what will happen right now means that this chromosome is carrying only this much portion and this chromosome carries this much portion. So for this particular flower color gene, purple color was dominant over white color. But since the purple color allele is not existing right now, the white color allele will come into action. Okay, so this particular condition is called as pseudodominance, and the condition of the chromosome is called as hemizygous. Hemizygous means here only one allele, the gene is represented by one allele. So for this particular condition is called as what I was saying you here is if the particular condition is deleted means that condition will be called as hemizygous condition, and the expression will be called as pseudodominance. That in spite of being a recessive allele, it will come into action in the situation where the dominant allele is lacking in the genome. Okay. Then genotype means it is the expression, if it is a genetic expression of an organism, like what are the alleles, what are the alleles that are present in the particular individual. If you type of the particular individual, okay, like what are the genes present, what are the gene and allele combination present in the individual, that is called as the genotype. And this term was proposed by Johansen. This question has been repeatedly asked in the exam, and the year is also important, so you need to remember all the terms whichever I present means written here, all the terms given by which scientist, and if possible, so for some important terms you need to remember the years also, like gene, genotype, phenotype, everything. For that you need to remember the years also. Okay, so term was proposed by Johansen for heredity and genetic constitution of an individual. So for plant height you can tell that capital T, capital T, small t, small t are different sort of genotypes. Okay, genotype will always and always be the expression of diploid individual. Whenever means whatever the individual is, if the individual is itself haploid means you will represent the genotype as one allele. If the individual is diploid means you will represent the individual as two alleles, because it will carry two allele. It can be anything, capital T, capital T, capital T, small t, anything. If the individual is diploid means for a particular character you will represent that as three alleles together. Okay, so for that, for plant height, the genotype is like this. If you are talking about the genotype of the whole individual, so it will be like representing all the traits. Means for plant height, what it is, for skin color, what it is, pot shape, what it is, all the genes you have to put together, and for all the genes separately, you have to write the whether it is homozygous, heterozygous, or what. So that will be considered as the genotype of the particular individual, right? Then coming to the next thing, that is phenotype. It is the observable characteristic of an individual. Means what you see physically with your naked eyes, that is a thing that is called as a phenotype. Means actually what is expressed in the plant. Okay, this term was coined by Johansen in 1909. Okay, though the phenotype is determined by the genotype, but certain genes do not express for a number of regions. So genotype and phenotype of organism may or may not be same. Why they're telling like this means, there are genes present in the individual, but because of certain region, that region might be what? That region might be some sort of epistatic interaction, some sort of environmental influence, some sort of photosensitive interaction, or thermosensitive interaction, or that character might be a threshold character. Like yesterday I explained, like threshold character is a character which will express itself only when the particular set of condition is available. That those characters are called as threshold character. So there might be a numerous reason why genotype is not, means phenotype is not a exact expression of the genotype. So genotype and expected phenotype may vary. Okay, that's why they're telling that genotype and phenotype of organism may or may not be same. Okay, then parental generation, because we'll be seeing so many of the crosses, so you need to know what is parental generation, what is F1, F2, and all those things. Okay, so parental generation, we'll call that as P. It will be designated as P everywhere, and we'll be calling the, if there are two parents, we'll be calling one as P1, one as P2. Okay, like this. So the parents which are used in a cross are said to be the parental generation. And essentially, when we are going for hybridization program, there is a basic requirement that parent need to be in cell pollinated crop, they need to be inbreds. In cross pollinated crop, they need to be in cell pollinated crop, they need to be pure lines. In cross pollinated crop, they need to be inbreds. And what is the similarity between them is both of them are highly homozygous. Okay, in case of inbreed, you can see some sort of residual heterozygosity remaining, and both, but still we can consider that as almost homozygote one. So for cross pollinated and cell pollinated, we'll directly use the inbreds and pure lines as parents. Okay, and they need to be very diverse to each other, so that we'll get a good amount of heterosis in the progeny. Okay, so that is regarding the parental generation. Next is F1. F1 stands for the term that is phylus or phyla, means the Latin, it is there from this Latin word phylus, which means son or daughter, means your progeny, immediate progeny. So F stands for that. And so F1 is what? It is the cross product of, immediate cross product of, cross of any two parents. Okay, so when you are crossing F1 with F2, the progeny which is coming out of this cross is will be called as F1. Okay, so a P1 and P2 both are true, means a true breeding type, because they are homologous in nature, and the F1 arising from them will be P1 and P2 are different in genotypic constitution, right? So F1 will be heterozygous one, and F1 will be homogeneous. So what I mean by homogeneous and heterozygous, can anybody tell me? F1 is homogeneous and heterozygous. What do you mean by homogeneous and heterozygous over here? Yes.
Ma'am, homogeneous means all the individuals will carry same genotype, and ma'am, heterozygous means the in genetic constituent one will be one allele will be dominant and other will be recessive.
Yes, means homogeneous means all the plants will carry similar genotypes, and all the plants will look alike. If you see them phenotypically, all the plants will look alike. Means there is very less amount of difference you can observe phenotypically also. And even if there is some sort of difference are observed in the phenotype, that is basically due to environment. If you are giving the proper kind of environment, similar environment to all the plants, there will not be any sort of variation in the phenotype. So that is what called as homogeneous. Okay, so F1 will always and always be homogeneous. And heterozygous means, as I said, it is the condition, like I already said, it is the condition where both the alleles will not be similar. So one allele will be dominant in nature, one allele will be recessive in nature. Okay, so that is the condition. That's why remember the fact that F1 is always and always homogeneous, heterozygous. What about the P1 and P2? Can anyone tell me what is the whether they will be homozygous, homogeneous? What is the condition of them?
Homogeneous, homozygous.
Yes, P1 and P2 because they are either they are pure line or inbreds, so they will be homogeneous and homozygous. So they are already product of several generation of selfing. So because of that reason, they will be quite homogeneous, and their all the their everything has been fixed. Means they are became the homozygous condition. So always and always remember that P1, P2 will be homozygous, homogeneous. F1 will be homogeneous and heterozygous. Okay. After this, whenever you are going for selfing, there is a symbol we denote for selfing. Anyone can tell me how to draw the symbol for selfing? When we are writing the cross, no, like this P1 cross P2, then we'll write like arrow, we'll give a arrow mark, we'll write F1. Then for selfing there is a symbol. Anyone know what is that? No. So see, always and always remember, when we are going for selfing, we'll make a circle first, then we'll put a cross mark inside this. This is a symbol for selfing. So without writing the thing that here we are doing selfing, simply this symbol, draw the symbol, circle and a cross inside the circle. That symbol denotes that you are doing the selfing event. Selfing means what? What do you mean by selfing? Yes.
Self self pollination.
Selfing means self pollination. Uh, you cannot say exactly self
एक ही पेरेंट
pollination.
को मैं मेल और फीमेल दोनों कंसिडर करके और क्रॉस
Yes,
Yeah,
करें.
sir.
yeah. Self means self pollination in the sense that we are pollinating with the self own flower itself. So same plant will be considered as male, same plant will be considered as female. So that is the case. Okay, means we are pollinating. But if you just exactly tell the word that is self pollination, means there might be confusion that the original plant was cross pollinated. So you cannot use the term exactly self pollination, but you can tell that we are doing, means we are pollinating with the same flowers pollen. Yeah, somebody raised hand. Yes, please pick up. What's your doubt?
मैम होमोजीनस और होमोजाइगस में क्या डिफरेंस हुआ?
ओके, मैंने अभी बताया तुम्हें, तुमने क्या समझा वो बताओ पहले, मैं फिर बताती हूँ.
मैम मैं होमोजीनस और हेटेरोजाइगस तो समझ गया, बट होमोजीनियस और होमोजाइगस में क्या डिफरेंस हुआ, ये थोड़ा बार बार कंफ्यूज कर जाता हूँ मैं.
There is means basically, see, to tell you, there is a plot. Okay, there are 10 plants right now. How many plants I can draw? Okay, 6 plants I can draw. There are 6 plants. So the condition is that if the 6 plants have, if the 6 plants have similar genotype. Similar genotype means they will be producing the phenotype almost similar. Forget about the environmental factor. So they all will be looking similar, right? In this plot, that wherever this condition is satisfied, you will call that this population is homogeneous. 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 Genus means always you will see the how the plant is looking. Okay.
फेनोटाइपिक कंडीशन.
Phenotypic condition. Exactly. Phenotypically.
ओके
Then the homozygous or heterozygous, that is genotypically you will be telling. So if the plant is means self pollinated, and the process here you need to see how the particular population is developed, whether it is a self pollinated one, it is a cross pollinated one, or it is developed with homozygous. Here means inbreds are developed with the help of, it is a cross pollinated product of cross pollinated plant only, but they are developed with the help of several generation of continuous selfing. So after that, the end product will be homozygous in nature, means all the alleles, all the genes have same allele at both the chromosomes. at the homologous chromosome, same alleles will be there. So homogeneous means genus thing. Whenever you are telling homogeneous and heterogeneous, that will be telling about the phenotypic expression of the plant, whether all are looking alike or not. And the heterozygous or homozygous, that will be the genotypic constitution, means whether similar alleles are present in the both the chromosome or different alleles are present in the both the chromosome. That you need to see. Okay, that is the difference between
मैम
homozygous. हाँ बोलो.
तो मैम अगर केस हाइब्रिड का आता है तो वहां पे तो हम फिनोटिपिकली सुपीरियर को चूज करते हैं या फिर बैक क्रॉस फॉर एग्जांपल करते हैं हम, तो वहां पे तो मैम होमोजीनियस होमोजाइगस P1 P2 कंडीशन फॉलो नहीं हो पाएगा, मतलब वहां हेट्रोजाइगस हेट्रोजीनियस हो जाएगा.
कहाँ पे F1 में?
हाँ, सपोज अगर हम बैक क्रॉस प्रोग्राम कर रहे हैं तो
हाँ
एक रिसिपेंट पेरेंट होता है, एक डोनर पेरेंट, तो डोनर पेरेंट
हाँ
सपोज वाइल्ड है तो उसका कैरेक्टर कुछ
हाँ.
और होगा, तो वो तो हेट्रोजीनस हो जाएगा.
नहीं, whenever we are going for back crossing, even no, the back cross parents have to be in inbred condition only. Then only we can take them. Okay, we have to bring the particular parent. We have to bring the particular parent to an inbred condition. Then only we can
ब्रिजिंग
use them for crossing program. Okay.
ब्रिजिंग करते हैं मैम.
Yeah. So whatever the condition is, F1 will be always and always homogeneous and heterozygous. After backcross, you will get the segregated product. Okay, that will not be homogeneous. Then you will get two types of product, two or three types of product based on which particular genotype you are using. So we'll see very soon. We'll go for the backcross breeding method. There you will see what are the composition of the backcross progeny. Okay.
ओके मैम.
Okay. Is it clear then?
यस यस मैम.
Okay. So next is the character. So what is a character? Is it is the morphological feature of the individual. Okay. So character can be morphological, anatomical, biochemical, behavioral, any feature of the organism which will be denoting to a particular, means which can be quantified. So that sort of thing will be calling as character. Okay, like seed shape, cotyledon color, plant height, or anything which we can quantify, which we can observe. Okay, either morphologically, either biochemically, or by any means, that will be called as character. Then hybridization is the process of crossing of two pure lines or inbreds to produce the progeny which is having superior traits, superior performance to both the parents or any one of the parent. Okay, and the getting the heterosis or hybrid vigor is the main purpose of the process of hybridization. So what is hybrid? Then hybrid is the product of crossing between two genetically different individuals. Okay, either they may belong to same genus or they may belong to same species. Okay, or any genera, between two genera. So depending on that, if we are crossing between two species, sorry, if we are crossing two plants between same species, it will be called as intraspecific. Intraspecific. Intra means within. Inter means between. Okay, always remember the two terms. Intra means wherever this term you will get, no, intra. So that is within. And inter means between. Okay, so intraspecific means within the species. Within the species means there are within species there are several varieties are available. So you will be crossing between means there are within species, within varieties you will cross. Okay, so that is intraspecific. Then interspecific means between species. Okay, inter means between. So between species you will cross. So here the genus might be same. So between species and the genus might be same, like Gossypium hirsutum cross Gossypium barbadens. If that cross you are taking Gossypium and Gossypium are same for both the cases. So genus label it is same. Gossypium hirsutum cross Gossypium barbadens. These are the two species in the cotton family. So here the genus is same but the species is different. So it is interspecific. Then if you are changing the genera itself, that condition will be called as intergeneric. Means genera itself is different. Means Gossypium we will cross with something else. Okay, Abel moscus if you are crossing. Both are belonging Gossypium hirsutum, cross Abel moschus. Abel moschus means which plant? Yes.
भिंडी, भिंडी ओकरा.
Yes. Bindi. Ma'am, interverator intera. Uh, Ankit, please pick up exactly what you wrote. The spelling is little bit mistake. So it is meaning two different types.
Intra species can also be called as intervarietal.
Yeah, you can call. But there that means it can be called, but the you have to see the condition whether you are crossing two different varieties or not, or the varieties might be very close to each other.
हाँ, that
So
will be
basically,
again
we can call that.
intra.
Basically, we can call that because within species, varieties are only available. So we can call that in a broad sense. But very precisely, if you want to say for any intervarietal thing, you have to check which two parents you are using, whether they are different variety or not. Okay. So basically, broadly, you can call intra specific as intervarietal. Okay. Okay, so that is the thing. Then coming to dominant character, recessive character, I already explained. So coming to the next thing, that is monohybrid, dihybrid, and polyhybrid. Monohybrid is a cross where the two parents are differing only one trait. Means the parent are same for everything, differing only one pair of contrasting character. Okay, so here single character is considered at a time. Then in dihybrid cross, you have two characters. Okay, so two characters considered here. And polyhybrid cross means here many characters are differing in the two parents. Okay, then there are another term that is direct and reciprocal crosses. What is direct cross and reciprocal cross? Anybody have idea?
I mean reciprocal cross. The if suppose in first generation we
First
selected
tell
one
about
plant
direct
as
cross.
डायरेक्ट क्रॉस में हम सिंपली एक को मेल दूसरे को फीमेल बना के क्रॉस कराते हैं, जो जनरली करते हैं हम.
हाँ.
और रेसिप्रोकल में हम सपोज एक को मैंने फर्स्ट जनरेशन में मेल माना था, उससे बाद में फीमेल कंसिडर करके और फिर क्रॉसिंग कर.
Okay. Guys, I want everyone to participate. Only one or two persons are participating. This is not a good thing. Okay, everyone should participate. Whatever you know, don't feel ashamed. If it is wrong, also it's okay. You need to participate, or else you will be sleeping halfway in the class and finally you will be leaving the class. So, whom I am going to talk to and whom I am going to teach. So please participate. It will be make your yourself active also, and it will means give me energy to teach also because it is already late, right? So direct crosses means p1 and p2. So in direct cross, let's say there are two parents p1 and p2. In direct cross you are considering p1 as female, okay, and p2 as male. So if that this is the condition p1 and p2, female and male, in the reciprocal cross what you will do, just alter the male and female type. That is the p1 will be here in the reciprocal cross. What will you do? P1 will be used as male and p2 will be used as female. Anybody has idea what the symbol I am writing? This is a symbol. This is the circle you have to make, and a plus symbol will be there. This is the symbol for female. This is this one circle will be there. One arrow and the one corner, that is the symbol for male. Okay. This is a symbol for male and female in any of the cross. Right? Anyone knew previously?
Yes, ma'am.
Okay. Even if you don't know or know, it's okay. And by convention, whenever you are writing any cross, you have to write the first the female parent, then the male parent. Okay, so for if the direct cross you are writing like this, means reciprocal cross, how you will write it? P2 cross P1 you have to write. Okay, why? Because P2 you are now using as female. So means whenever it is written like P1 cross P2, simply they have not mentioned anything like male female. So you have to consider that P1 is the female, P2 is the male. And whenever P2 cross P1 is mentioned, you have to consider that P2 is the female, P1 is the male. Okay. And why we do the reciprocal crosses? We do the reciprocal crosses to find out whether both the parents are making an equal contribution or not. If the both the parents are making it equal contribution, means there will be no change Means simply that if both the parents are giving 50% of their genome via gamete, so there should not be any difference in the reciprocal crosses. If we get any sort of difference, so we'll be calling that them as basically the we see the differences when we see any sort of maternal inheritance or cytoplasmic inheritance. If the traits are governed by the cytoplasm of the organism, that time only we see some this sort of variation in reciprocal crosses. Why? Because the female parent only contributes the cytoplasm to the progeny, not the male parent. Okay, and the contribution of the male parent or pollen is very very negligible. So if you are locating any sort of difference in the reciprocal crosses, directly you can means you may conclude that those sort of traits are basically because of the maternal inheritance or cytoplasmic inheritance. Okay, then coming to the next term, that is backcross. Backcross is the process where you will be crossing the F1 with any one of the parent, any one of the parent when we are crossing either P1 or P2, irrespective whether it is dominant or recessive. No need to look for anything. Simply you are crossing with any of the parent, that is called as backcross. Whenever you are crossing the F1 with recessive parent only, that condition will be called as, yes,
Test
that
cross.
Test
condition will
cross.
be
Test
test cross.
cross.
Exactly.
Test cross,
Good.
test cross.
So that condition will be called as test cross. So backcross is the quick way to make the desirable homozygote individuals. Why? Because with the every generation of backcross, you will get 50% of homozygosity will be increased gradually with each generation of backcross. So because of that reason, backcross is a quick way to make a homozygous individual. But if you want 70 or 80% of homozygosity, it will be very quickly achieved. But thing is that to get 100% homozygosity, it is very tedious that you have to go for several backcross generations. Means BC5, BC6 means backcross 5 to 6 generation of backcross you have to take, and you have to very carefully observe for the plants. Then test cross I already told you. So what is the use of the test cross? To determine the genotype of the unknown individual, that is whether it is homogeneous, homozygous or heterozygous. Here the condition is that if you are crossing here, here the cross is not given. Okay, so when you are crossing the hetero means F1, that is in capital small a condition with the recessive parent. So this is the recessive parent. If the progeny is segregating, here the what will the progeny? Here you will get, from here you will get capital A and small a, and from here you will get only small a. So the progenies will be capital A small a and small a in 50%, means 1:1 composition. If this is the combination comes, means you can tell that this is the condition is like this is the original parent wise, means original plant. Here this plant's genotype means the genotype of this particular plant is unknown previously. If here 1:1 condition comes, means you can tell that this the individual original genotype was heterozygous one. Suppose the condition is like capital A capital A, the original genotype is homozygous one. So what it will produce? It will produce only one type of progeny. There will not be any segregation pattern. So at that time you can tell that the original genotype was homozygous one, not heterozygous. Okay. So that is the condition. That is how the test cross is used. Then heredity is the thing that is the it denotes the to the thing that transmission of genetic character, means how the characters are transmitted from one generation to another generation. This is a famous statement given, that is like begets like, means how the similar means humans are always giving rise to humans only, not chimpanzees or monkeys. So every time whenever human or any plant or any animal they are going for reproduction, there the progeny will be like to them. Alike means there might be some sort of morphological difference between them, but they will have two legs, two eyes, or two hands, and one nose, one mouth, like that. Okay, so basic structures and everything will be similar only. So like begets like, so the behind that thing, the cause lying here in the genetics is, that is the heredity. Okay, and this is another term, that is Punnett square. Punnett square is a diagram which is a square sort of diagram which was given by Punnett, scientist Punnett. Before his name, it is given the name, that is Punnett square. This diagram actually used to predict the genotype of a particular cross. So what you will do here? Here you have to place the particular crosses, means the Means here male, this is the male, this is the female. So for, sorry, this is female, this is male. So you have to put the female gametes over here, male gametes over here in both the arms, and you have to put the crosses and write down the particular genotype just by multiplying. Say here capital A is there, capital A, so here it is coming capital A, capital A, capital A, and small a is there, so you just multiply. Means what, if here any frequency or anything is mentioned, means here it is half, let's say it is half, it is half. Why I putting half up means originally it is one, no, original frequency is one. So from here to sorry, not this one, this one. So from here there is half probability that it will produce capital A genotype of capital A allele in the gamete, half probability that it will have small a allele in the gamete. So that is the probability is half half. Similarly here also half half. So whenever we are going for any sort of calculation, what will be, you do, here you have to multiply the frequencies. So what will happen here, over here, half cross half into half, that is the frequency of this particular thing, that is 1 by 2 into 1 by 2 means 1 by 4. Similarly you can see this is 1 by 4, this is 1 by 4, this is 1 by 4, this is 1 by 4. Okay, so this one will be one, these two are similar, so it will be 2 by 4, this is 1 by 4 and 1 by 4. That's why ultimately we are writing this as 1 by 4 is to 2 by 4 is to 1 by 4. That is, first one is representing capital A, second one is representing capital A, small a, third one is representing small a, small a. Okay. So this sort of multiplication and these things will come in the various problems associated with Punnett square. Okay, it was named after the scientist, that is Reginald C. Punnett, who devised this approach. Okay. So so these first three slides are very important for your examination point of view. You need to be very clear regarding these terms because they will ask directly from these terms. Okay, so that's why all the creamy parts I put in the first only. Okay, let's get into the actual Mendelian thing. So there are several theories given by the pre-Mendelian scientist. So apart from this theory I have listed, if you get any theory, just add to your notes. That will be very helpful. Means very often they don't ask from this any questions. Important ones I have covered, but there are several theories which is not that much important. But still the name and the scientist name, that is only important. For all this theory, little bit you need to know, that's it. And the first one is moist vapor theory, that was given by Pythagoras. He believed that some sort of ultimately giving rise to the progeny. So that is a very vague kind of theory that was very soon eliminated by the scientists. Then theory of preformation was given by Swammerdam. So there he told that some sort of previously formed organism will be there. So that is not the case also. So here just you need to know that who has given the particular theory. Okay, that is the thing. Then the next one, yeah. So this thing, this pre-formation theory was replaced by the theory, that is theory of epigenesis. Okay, theory of epigenesis was given by Wolf. So which one replaced this one? So in between this means a little bit problem with the order I have mentioned over here. So this theory of preformation, when it was discarded, the theory of epigenesis took its place. That was given a Wolf. So Wolf has and epigenesis theory was further refined by von Baer. Another scientist was there. Okay, and he has mentioned that some sort of homunculus is present in the sperm and egg, and those sort of things are further going and they are differentiating into individuals. So that those are much not much important ones. Still epigenesis is one of the universally accepted thing that everyone has accepted. So this epigenesis is little bit important, that scientist who has given. And the theory of germplasm, that is Weizmann has given. And the famous experimental material for Weizmann is On whom?
Rat का tooth
Rat.
काटता.
He has cut the tail of the mice, that is for 22 successive generation, and he wanted to see that whether after cutting the mice tail, whether it is acquired or not. So he clearly see that it is not acquired. Every time after cutting also the progeny will have the tail. So that was actually the theory which discarded the theory of acquired character, which was given by Lamarck. Lamarck said that whatever means all the physical fitness, intelligence, what one individual acquires, that will be carried forward to the progeny. But that is not the case. So it was discarded by the theory, that is theory of germplasm. Okay. Then next thing is that theory of pangenesis. Theory of pangenesis was given by Darwin, where he has said that some sort of pangenes or gemmules will be there, and this each organ will be producing some this sort of individual small molecules, and ultimately these from all organs they will be transported to gonads and they will be giving rise to the organism. So that was discarded again, and that was discarded by this Weismann's theory. Okay. Then another theory was there, like theory of organic evolution. um so it was a little bit means it had a sound background on its own. um and that's it regarding the previous theories. If you get any other theory, you can take them also. So coming to the facts regarding Mendel, whatever facts there from the Mendel's birth to death, everything, where he was born, date of the date of birth, date of death, when he took with sort of study, what he did, how many characters he chose, everything regarding Mendel is important. Why? Because he's the father of genetics. So anything can be asked in your exam. Okay. So be careful. So whatever data you can gather regarding Mendel, that will be useful for you always and always. Okay. So first thing is Mendel date of birth. Anybody know when Mendel took birth? No. It is 1822. Date of birth. Anybody? Any idea? No. So write it down. It is July 22, 1822. Okay. So in Wikipedia and some other pages, you may find the date like July 20 also, but it is original date is July 22. Okay. So that is the day, and he was born in Austria, that is Brunn. And after that, he has given the law of heredity, and he worked with the, here I have mentioned only the important points. He worked with the garden pea, that is Pisum sativum, and he took 7 pairs of contrasting characters. He demonstrated the theory before Natural History Society of Brunn. This society name is very important. I'm telling you, before whom he has explained the theories. He has explained all this thing before the Natural History Society of Brunn. Okay. That is at the meeting, when the meetings were held in 1865. The exact date is February 8 and March 8. Those two dates you need to remember. And the first paper presented by him was Experiment in Plant Hybridization. And he also presented another paper in 1867 on heresium. I'm forgetting the exact title of the heresium paper. That is just a second, I'll tell you. His second paper was published in 1870, not 67. That was on Herasium hybrids obtained by artificial fertilization. That was the paper title, and it was later on. Both the papers were translated by Batson in 1902, first paper and second paper, to English. Original paper was written in German language, and for second paper and first paper, both papers are important. But conventionally, you can find this paper, that experiment in plant hybridization, most in your textbook. Second paper is not mentioned, but he had two papers. Okay. And the next important thing is that he was the first person to apply the statistical analysis to genetic studies. Okay. And he died in 1818. When he died?
1884.
Yes, he died in 1884, and the paper was rediscovered in 2000, that is 16 years after his death. Okay.
Ma'am, that
Ma'am,
is
1912.
1900, not
1900.
2000.
Sorry,
1900
sorry,
is
sorry, 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 sorry. Sorry, this is 1900. Thank you. All are awake then. Okay. So the paper was rediscovered in 1900 by three scientists, that is Hugo de Vries, Carl Correns, and Tishmark in Austria. So here you need to remember the three scientists' name and the country of their origin. So you can remember it like this, means Hugo de Vries is from Denmark. It is sometimes called as Holland also. Okay. So Hugo de Vries is from Holland. So H for H. Hugo de Vries is from Holland. And
CGTA.
this is CGAT. So ATGC and HH. So ATGC you normally know in DNA, it is there. C for Correns and G for German. Tetramark for Austria. So H for H, that is Hugo de Vries for Holland, and Correns for Germany, and Tetramark for Austria. That's how you can remember. Okay. Another thing you might be knowing or might not. Anybody can tell me on which crop de Vries has worked and rediscovered while rediscovery of this law? Which crop he has worked with?
Mirabilis Jalapa.
Yes.
Ma'am, which Hugo de Vries on Oinothera americana.
No, he didn't work. He worked several things in Oinothera lamarckiana, but for rediscovery, he worked with a plant that is called campion. You can see the particular flower in the book Simons and Oster. There is a pink color flower. Um, okay, wait a second. And Carl Correns has worked with P
Ma'am,
and Mage. Okay.
Hmm.
But Tessemax, which crop he worked with, I didn't find.
ma'am, P,
I'll
ma'am.
P.
That's for
4
Okay.
o'clock plant, ma'am, sir. Mirabilis Jalapa.
Okay, I'll check that and confirm. Okay, I didn't find it. So that's why. Wait a second.
First, ma'am, he worked on Garden P only, but later on he moved to Mirabilis Jalapa, I think 4 o'clock plant.
Tessemax, you are telling, no?
No, ma'am, Carl and Corin.
Carl Correns.
Yes, ma'am.
But there is a book named Origin of Genetics that is written by Kurt Stern. Okay, I was going through that book. He is the one who actually collected all the papers of Mendel, and all the papers, all the letters Mendel has written to Nageli, and all the discoveries, means original report of the discoveries. So according to that book, that source, this is one of the very old book. So according to this particular book here, what I found is Kurtzton, you might be knowing, he was the first to give the proof for what? Kurtzton. There is a famous experiment. Yes. Kurtzton, anybody? Any idea? Who is the scientist? No? Kurtzton, he has given the experiments proof for cytological proof for crossing over, that chromosomal segment exchange occurs. Okay. Kurtzton has proved that in Drosophila, and Clayton and Barbara McClinton have proved that in maize. Okay. So that Kurtzton's book I was just going through, there it has given that Hugo de Vries has used this particular thing, that is campion flower, and Carl Correns has used this is the
Mirabilis.
flower. Can you see this? This is the plant. De Vries experiment with flower color and foliage types in the varieties of campion. So this is the flower where he actually given rise to your monohybrid cross and dihybrid cross. Okay, this is the plant, that is campion. Okay, from here I saw this one, and from that book, that is the origin of genetics, Mendel's papers and everything, translated ones, original translations are there. So there they have mentioned that Carl Correns has worked with P and mage. Okay, regarding Mirabilis jalapa, I have to see, I'll confirm later on. Okay, and Tessemac one I didn't find, that one also we need to see. Okay. Okay. Then regarding rest of the Mendel's history, like Mendel, when he became a priest, when he studied, that those things are not that much important. Still, you should have a better idea regarding them. Okay. Then coming to next thing, that is Mendel's postulates. Okay. So before going to this, I want to all of you to answer, that is what was the mistake made by the predecessors of Mendel, means why they were not getting the actual results which Mendel got. What was the
मैम
problem with them?
वो रिपोर्ट नहीं बनाते, प्रॉपर रिकॉर्ड नहीं रखते थे,
Yeah. Next.
स्टैटिस्टिकल मेथड यूज किया था इसमें, क्योंकि मैथमेटिक्स बैकग्राउंड।
Next.
Mostly they studied on the quantitative characters.
Yeah, because they studied the quantitative characters, they were not getting the exact segregation pattern which Mendel got. They were always getting the continuous pattern of variation. That's why they were not coming to the conclusion. Yes. So
लॉन्ग
these are some
ड्यूरेशन,
of the. हाँ बोलो.
बाकी साइंटिस्ट में लॉन्ग ड्यूरेशन प्लांट को यूज करते थे, ये शॉर्ट ड्यूरेशन।
Yeah, long duration plant, when you are using long duration plant, they are very difficult to, you have to wait for very long time for generations to advance. So that was one of the problems. Okay, that is also a problem, but basic problems with them were they were not taking the good records of their things. They were not able to classify all the things, and they didn't use the statistical way which Mendel has used actually. And another thing is that Mendel always and always took the characters very precisely. They didn't look at a particular character at a time. They were taking all the characters simultaneously, so they were getting confused. Okay, but Mendel took the observation, and for one of the character, means what, whenever he was going for crossing, he only focused on one character or two character. So that made half of the job done. Means that made the analysis quite simpler. Okay, that was the major reason for Mendel's success and his predecessors' failure. Okay,
Now, how many years did Mendel took to complete his experiment?
I guess 7 years. From 57 he started. It is the mentioned in some one of the books. Where it was mentioned. Somewhere I read it is 7 years. Okay. From 57 he started doing his experiments. 7 years means 64 he finished, and 65 he published his book. That paper he published. So it is 7 years. Okay.
Okay.
And Mendel has initially took how many varieties? Yes, that also you need to know. Mendel has used initially 34 varieties of pea, but later on, depending on the availability and the crossability and all those things, he returned back to 22 varieties. So finally, he used only 22 varieties for his hybridization experiments. Okay. And okay, so this is regarding that thing. And why you chose the pea plant as one of the experimental material? Because it is very easy to cross, it is a very short heighted plant, characters were quite distinct, and the what generation in 1 year they can forward 2 3 4 generations even. So it is a pulse crop, so there is no easily crossable, means flowers are quite bigger one, and it is having what cell pollination mode, so easily you can remove the anthers to make the emasculation and pollination. That is relatively easy thing. And so all these are the traits. Okay. And Mendel's characters are present on the seven characters are present on the 4 chromosomes. So exact chromosome number also you need to know. So can anyone tell me which are the chromosome numbers Mendel used?
1457.
1457.
So that will depend on which particular chromosome, that I will send. Okay. Means exactly right now also I am not recollecting the exact which trait was present in which particular chromosome, that I will send. Okay. So that one also you need to know. Because Mendelism is, means from Mendelism, everywhere Mendelism is one of the favorite parts wherever they go for examinations. So you need to be very thorough for all these facts and figures. Okay. Then next thing is that the term genetics was coined by Batson, and when he coined the term genetics,
1902.
1905.
1905, not 2.
1901.
1905.
Okay, okay, there is very big confusion then. I'm very poor at the years.
मैम
I'm sorry to tell you.
1901 में मैम बैटसन और पुन्नेट जर्नल ऑफ जेनेटिक्स स्टार्ट किया था उन्होंने शायद।
Yeah,
और 1905 में टर्म जेनेटिक्स फॉर्म किया गया था।
1902, I guess. Wait.
मैम 5 होगा क्योंकि मैम पहले मेंडल में पहले मैंने फादर ऑफ जेनेटिक मेंडल को बोला गया था, उसके बाद ही जेनेटिक्स टॉम आया था। 2 साल बाद आया, 2 या 3 साल बाद आया था।
मैम 1905 है।
Hmm.
मैम बीडी सिंह पुन्नेट सिंह को 5 में।
Okay. I'm so sorry. I'm little bit bad with the years. Okay. So that's why.
वॉइस इज ब्रेकिंग।
So okay, don't quarrel. We'll be coming to a conclusion. 1902 or 1905. I whatever, as far as I remember, it is 1905.
इसी
Still,
साल
I will not
इसी
speak
साल मैम
anything.
लिंकेज भी डिस्कवर किया गया था पुन्नेट के साथ मिलकर।
Yeah, 1905. Okay, it is 1905. And then the next thing is Okay. So that was 1905, and that year only linkage was also discovered. Batson and Bonnet, that was correct. So coming to the Mendel's postulates, Mendel has given the four postulates basically. That was different version of the genes account for variation in the inherited characters. That is different version of the genes means alleles. Okay, they are giving the variation in the characters. Then for each character, an organism inherit one basic generalization initially Mendel has made. Okay, there is nothing much to read here. If there are two alleles, different different one is there. The dominant one will be fully expressed, while other one will not have any individual appearance. That is the recessive allele, and the two alleles of the each characters are separate when the gametes are created. That is Mendel's law of segregation. Okay, so how Mendel's pea plants are helps us to understand the genetics. These are the cute what garden pea seeds. So these are the traits, pea traits Mendel has originally took. That is flower color, seed shape, seed color. So you need to remember all the trait names basically. Okay, like which characters he has taken and which one not. Okay, sometimes they used to put like pod shape, pod color is there, no, they will put something else, means seed shape they will put, seed shape is there, seed shape is there. Okay, means flower shape or anything they will put, simply means altering the terms, shape, color and height, position. So they will alter something with flower, seed or pod, and they will put a option. So that thing you need to be very careful regarding. Okay, then why Mendel did work with pea plants, so I already explained you. So pea plant is easy to work with, so simply you have to open up the flower and remove the anthers, and then you go for crossing and it will produce the progeny. Then what is the monohybrid cross? I already explained that two plants with their capital PP and small pp. They are the two parents, and here you are crossing and going for the heterozygous one will be the F1, that is the first generation of the progeny. If it is self-pollinated means PP will be crossed with PP, capital will be small p. Okay, and here like I said you have to put the female one over left side and male one over the right side, and you will be whatever the Punnett square will be formed, that will show you the progeny genotypes. Okay, so this is the condition. Here you can see, so wherever the capital P is present it will express itself. So in these three conditions the flower will be purple in color, and this particular condition the flower will be white in color. So here it is giving rise to the phenotypic expression, that is 3:1. And the genotypic constitution, if you see, this is 1, this 2 will make 2, and this is 1. So here the genotypic constitution will be 1 2 1. So this is the basic thing you already know. So in monohybrid cross, always and always your genotypic ratio will be 1 to 1, phenotypic ratio will be 3:1. So from monohybrid cross only we are getting to the inference regarding the law of dominance, which states that every gene has two alleles that can code for a particular trait, and one allele will be dominant. It will always show wherever it is present. One allele will be recessive. It will be masked by the presence of the dominant allele, and hybrid will always show the dominant phenotype. Okay. Then three important points regarding the dominance thing is that ranges from complete dominance to incomplete dominance or codominance. Means dominance can be anything. Okay, we cannot always say that dominance will be complete dominance only. So the range of expression of dominance may vary regarding based on the particular gene. And the dominance allele does not suppress or interact with the other allele. The recessive allele is simply not expressed by the cell. Means it is not that they are both are interacting with each other. It is not the case. Interaction will only come in the case of your epistasis. But to tell you simply that dominance we are calling that intragenic interaction, that two alleles in alleles interact, but here the interaction does not mean that here it will produce some product which is inhibiting that other thing, recessive one. It is not inhibiting, simply that it is producing the functional one, recessive allele is producing the non-functional one. That is the thing. So recessive allele is not able to express itself, and the dominant allele is able to express itself. That is the case. A trait being called dominant does not necessarily mean that it is more common in the population. Dominant means it is the it is the expression of a particular trait, but you cannot say that dominant means we cannot say. We have to see the frequency of the particular allele. There might be case that P means wherever we are going, sometimes you might be seeing P is the dominant allele, Q is the recessive. This sort of condition is also exhibited. The character might be, the allele might be dominant, but it not necessarily mean that it is more common in the population. Then coming to the law of segregation, so it tells you regarding the fact that the two alleles, they are present in the heterozygote, but they don't contaminate each other. They are inherited in the pure form in the progeny. Okay, that is the law of segregation. Means they segregate whenever they are going for gamete formation without contaminating each other. They are going for the that is the thing. And this was also the law of segregation was explained by monohybrid cross only. Okay, and there are basically three assumptions, four assumptions that are involved for law of segregation. The first one is that means all the gametes will be produced in equal frequency. All two types of gametes will be produced in equal frequency. Two types of gametes means these two, capital P and small p, they are produced in equal frequency and they have equal amount of survival, and there is random union between the gamete. Random union means here the capital P can mate with equal chance, it can mate with capital P and small p. There is nothing fixed that it will mate with this much amount or this much amount. It can randomly mate with anyone. Okay, and there is equal survival of the zygotes which are produced from this process. So these four are the assumptions which he took for the process of law of in segregation. So basically what is happening in law of segregation is that you might have seen that there are homologous chromosomes. Okay, in basically in your meiosis stage, in the metaphase you can see, in metaphase this is the condition. This is the condition for one chromosome, this is the condition for other chromosome. T is present for, let's take a particular trait that is let's say for plant height. Okay, so here capital T is present, here capital T is present, here small t is present, so definitely here small t is present. Okay, so what is happening in meiosis 1 is, in case of your metaphase 1 they will align at the metaphasic plate. Here the cell is there and they have aligned at the metaphasic plate. Now what is going to happen in the anaphase 1? What will happen? Yes, what things will segregate? This one chromosome will move to one pole, this one chromosome will move to one pole, right? So what will happen at the end of anaphase 1? So after anaphase 1, what is happening is, you can see this particular chromosome, this chromosome with capital T in both the side, in both the arms is in one pole, and another chromosome with small t with it is at another pole, right? So this is one cell and this is another cell, right? And from this cell again, this chromatid will segregate and ultimately four cells are being formed, four gametes are being formed, and four gametes will carry what? This one will carry a chromosome which is having capital T, this one will carry another chromosome which is having capital T, this one will carry another chromosome which is having small t, this one will carry another chromosome which is having small t. So here both capital T and small t were there in one one cell itself, but the thing is that in this particular condition the t capital T was only expressing small t was not expressing, but uh, but in this condition also they have their power, means the gene has not lost its originality, whatever condition that is kept like as such. Okay, without affecting each other. Okay, law of particulate inheritance, right? It is otherwise called as law of particulate inheritance because they do not contaminate each other. Okay, so this diagram tells you regarding the this is what happening in anaphase 1 and this is what happening in anaphase 2. anaphase 2. Okay, so that is the condition. So basically how the segregation means, how the law of segregation is actually working. The separation of homologous chromosome during the meiosis, that is a major region of working of means working principle of the law of segregation. That is because of the meiosis phenomena. At anaphase 1 the two alleles are getting separated, and at anaphase 2 the four gametes are actually being formed, and where we can see the two of the gametes are carrying one type of allele, another two are carrying another type of allele. That is the this condition will be there when the original individual is having heterozygous condition, that is capital T small t, right? And the time of segregation is that if here you can see there is no crossing over mentioned. If the crossing over event would have happened involving the centromere, then what will happen, the 1t and this capital T will be exchanged with a small t. So here capital T is there, here small t is there. So what will happen, even if the both the chromosomes are separated, the alleles are not yet segregated at after anaphase 1. So what the alleles will only be segregated when the chromatid segregation is occurring, that is in the anaphase 2. So if there is no crossing over event, so you can tell that allelic segregation is occurring at anaphase 1. If there is crossing over event occurring, in that the crossing over event including the centromere, then you can tell that the allelic segregation is occurring at anaphase 2, not anaphase 1. Okay, that is the important point you need to remember. So that is about the law of segregation. And this is the dihybrid cross. So here two traits are considered at a time, and here you can see they have taken the cross, that is round yellow with wrinkled green. Round and wrinkled are the regarding the seed shape, yellow and green regarding the seed color. Okay, so here the pattern of inheritance will be studied two traits at a time, and out of this particular dihybrid cross the law of independent assortment is given, which states that the inheritance of the particular one trait does not affect the inheritance of the other. So basically what it is telling is, whether in the progeny it is not fixed that every time from the round yellow and wrinkled green progeny will be producing round yellow together and wrinkled green together. It is not the case. Round can combine freely with yellow or green anything, wrinkled can combine freely with yellow or green anything. So any of the allele of one trait can pass on to the gamete with any of the alleles of other trait. That is what the law of independent assortment means in simple words. Okay, so what makes the two traits independent? So two traits are independent if they are inherited separately. Two traits are independent if they are inherited separately. In other words, the alleles for the traits, they do not physically affect one another. That is the thing that is called law of independent assortment. Okay, so this is the difference between so this is the difference between the Mendel's first law and second law. Here you can see that Mendel's first law is the law that explains the segregation of the alleles in the from the given locus into separate gametes. But this law tells that the traits inheritance is independent of one another. Means one trait inheritance is not affected by another trait's inheritance. The other thing is that it is called as law of segregation. It is called law of independent assortment, and the explanation is that during meiosis the members of an pair possesses an individual pass on to different gametes. Means any allele from the out of the allele pairs, only one of the alleles will be passed on to the particular allele, the particular gamete, not both. Anytime the gamete cannot carry both the alleles, always and always it will carry only one of the alleles. Okay, and they do not contaminate each other. That is the basic thing. And all the combination of the alleles assort independently and passed on to the offspring with equal probability. So they will be passed on independent to each other. There is no combination or there are no sorts of linkage that always and always red, that yellow one will go with round will go with yellow or wrinkled will go with green. Every combination you can see over this Punnett square. Round wrinkled with yellow, then yeah, round with green. Everything you are observing over here, right? So that is what the independent assortment means. Then it explains the behavior of all the chromosomes. like all the whatever the alleles are present in the whatever the alleles present in all the chromosomes, everyone will carry two alleles, right? And for all the chromosomes, and they are going to segregate independent, they're going to segregate when they are forming the gamete, right? But here only behavior of non-homologous chromosomes are mentioned. Why non-homologous chromosome means, here they have assumed that different traits will be present in different chromosomes, and they are not homologous to each other, right? That's what they have taken the assumption. That's why they are telling that this is applicable for a single trait. Here monohybrid cross is considered. Here two or more traits can be considered for independent assortment. Okay. So here you can see the this is one pair of chromosome and this is one pair of chromosome. So here you can see this is a metacentric chromosome and this is a submetacentric chromosome. So here one trait is present, here one trait is present. Here in the progeny, what they're telling that from this, whenever the gametes means any of the particular chromosome, means the both alleles will go to different gametes. So you can, here you can see in all the gametes only one of the two, means either this one is present, either this one is present. Okay, so that one you can see for all the chromosome. For this particular two chromosome, either this one is present or here this one is present, not both. So that is tells about the law of segregation. And here the independent assortment, you can see that this particular black color metacentric chromosome can pair with this white color metacentric chromosome or this white color band submetacentric chromosome or this one. Anything it can pair with. That is independent assortment it is telling. Okay. And in the F2 you can see the principle of dominance, that is only the dominant characters are expressing in themselves when the composition is like capital T. The recessive ones are expressed only in the case of homozygous one, not in the heterozygous. And these are regarding the various Mendelian traits. You can see the six-fingered character, the famous example Hrithik Roshan, as you all might be knowing. Then dwarfism also a Mendelian trait. Widow's peak, here you can see the widow's peak. Okay, means here the hairline will be somewhat sharpened in the mid forehead, and you can see the dimples. Then cleft in the chin, here you can see the cleft in the chin, and ear wriggling is also Mendelian trait. Apart from this, there are many other books. So that list is there in your CC. I'll send you that particular thing. Okay. Uh, okay. We'll deal with the exceptions of Mendel's law in the next class, and particular this slide also. Till now, any doubt you have, you can ask. And some few points are there, few objective and important points are there. I have noted somewhere else. I'll discuss in the next class regarding the segregation and independent assortment. Some important informations are there. Okay. Any doubt you have till now?
मैम इनब्रीड और प्योर लाइन में क्या डिफरेंस है?
इनब्रीड जो है वो डेवलप होता है क्रॉस पॉलिनेटेड प्लांट का प्रोडक्ट, और प्योर लाइन जो है तुम्हारा डेवलप होता है सेल्फ पॉलिनेटेड में से, और दोनों ही प्रोडक्ट होते हैं सेवरल जनरेशन ऑफ सेल्फ पॉलिनेशन का। ठीक है,
आइसोजेनिक होता है ये इनब्रीड?
सेवरल जनरेशन ऑफ क्या क्या
इनब्रीड मैम NIL
यस
कैटेगरी में आ सकता है?
NIL में in NIL you can see several inbreds are there together. So for the definition of inbred, you should not look at the thing that is NIL, because you will get confused. Because in case of NIL, they the individual lines in the NIL, they represent one inbred. Okay, that is the thing. मतलब NIL जो तुम बोल रहे हो ना कि बहुत जनरेशन ऑफ सेल्फिंग से ही बनता है, एक्चुअली वो करेक्ट है, पर thing is that NIL is a combination of different.
कहीं ना कहीं वो डिफर कर जाता है सेम ही कैटेगरी के
Yeah, they
दूसरे।
differ for one gene basically. Means that is the thing in case of NIL.
Yes, ma'am.
And they're all you can call that, because they are different for one gene, they are called as isogenic lines. Okay, So in NIS, all the lines are isogenic to each other. They are differing in one traits. प्योर लाइन और हाइब्रिड के बीच में बेसिक डिफरेंस इतना ही है कि दोनों ही होमोजाइगस और होमोजीनस होते हैं, बट प्योर लाइन जो है तुम्हारा सेल्फ पॉलिनेटेड का प्रोडक्ट है और हाइब्रिड जो है वो क्रॉस पॉलिनेटेड का है. और प्योर लाइन जो है वो खुद ही सेल्फ हो रहा होता है, तो तुम्हें उसको बनाने के लिए ज्यादा कुछ मेहनत करनी पड़ती नहीं है, मतलब तुम्हें बस सिलेक्शन करना होता है. पर इनब्रिड को बनाने के लिए तुम्हें कंटिन्यूअस मतलब फोर्स्ड सेल्फिंग करवानी पड़ेगी, क्योंकि इट्स नेचर इज means by default its nature is cross pollinated one. That is a diff thing. Okay.
मैम एक बार होमोलोगस क्रोमोसोम्स के बारे में बता दीजिए.
Homologous chromosome means, you see, in case of humans we have 46 chromosomes, right? So 23 chromosomes will be coming from one parent, 23 chromosomes will be coming from another parent. Parent in the sense that two gametes are mating with each other, sperm and egg, then only the zygote is getting formed, right? So in sperm 23 chromosomes will be there, in egg 23 chromosomes will be there. So ultimately they are going to form the 46 chromosome. So these 23 chromosomes are numbering as 1 to 23, 1 2 3 4 like that 23, and this one is also numbering as 1 to 23. So this chromosome 1 from male parent and this chromosome 1 from female parent, both of them are looking alike, but they may differ for the allelic constitution. Means in मतलब फादर में से जो क्रोमोसोम आया हुआ है उस पे जो भी जीन जहां पे होना चाहिए वहीं पे उसी सेम पोजीशन पे तुम्हें फीमेल के क्रोमोसोम पे मिलेगा, बट एलील वेरी कर सकता है. दैट इज यहाँ पे टॉलनेस का एलील हो सकता है, वहां पे ड्वार्फनेस का हो सकता है, दोनों सेम भी हो सकते हैं. ठीक है. So that is the condition of homologous chromosome, that one chromosome that is from father, one chromosome that is from mother. So both will together constitute the homologous chromosome. They are morphologically similar. Only the thing is that they may carry the different alleles. They carry the same set of genes. They are morphologically similar. Everything is same. The thing is that at a particular locus, the allelic constitution may vary for homologous chromosome. That's it. Okay. Hello. Yeah, in chat box somebody asked something. Okay, ma'am. Thank you. Anything else? Okay, thank you everyone. So tomorrow or day after tomorrow, I'll plan another genetics session for the rest of the part of Mendelian genetics. I'll tell you. By that time, what I want all of you, that this was a sudden class I didn't mention before, so you might have not gone through the chapter very well. So the important sections are, as I mentioned, you need to go through, and physical basis, which is mentioned in the basic book, that is important to go through. That will make you understand how the exactly segregation or independent assortment is. And if you find any sort of doubt, we can discuss later on, right? So thank you everyone. I'm ending the session here. Bye bye. Good night.
Thank you, ma'am.